Biochemistry 401 Lecture 36. Today we're going to talk about what can go wrong and how to fix it. Safeguarding the genome. We're going to talk about mutations and repair of those mutations, and then we're going to talk about recombination. Some things do not belong together, such as cytosine and adenine and guanine and thymine. But sometimes mistakes happen, and we're going to go over some of the things that can cause mistakes to happen, that can give rise to mutations. And so when problems occur with mismatching and mutations, generally speaking, it's all about that base. Hydrogen bond donors and acceptors. Normally, deoxyadenosine binds to deoxythymidine because of the patterns of hydrogen bond donors and acceptors, and the same with deoxyguanosine and deoxycytidine. These patterns of hydrogen bond donors and acceptors are complementary, and so they bind to one another efficiently. But when these patterns of hydrogen bond donors and acceptors are changed, that's when problems occur. In this diagram, we'll take a look at deoxyadenosine, and you can see that there's an amino group at position 2. This amino group has 2 hydrogen bond donors, and one of them makes contact with the hydrogen bond acceptor, oxygen, and deoxythymidine. It's a complementary pairing. The same thing for that nitrogen at position 1. It's a hydrogen bond acceptor, and so it accepts the hydrogen bond from the nitrogen at position 3 in deoxythymidine. That is a hydrogen bond donor, and so again, that is complementary. But when you change these patterns, when you introduce a hydrogen bond donor where it doesn't belong, or when you take one away, this is what causes problems. Some of the factors that change hydrogen bonding in deoxynucleotides are listed here. There's tautomerization, syn versus anti position of the base relative to the sugar. Water mediation. This is when 3's a crowd and the water molecule gets in between the 2 bases and forms its own hydrogen bonds between the bases. That can be bad news. Then there's oxidative deamination, alkylation, oxidative damage by reactive oxygen species, there's dimerization, and there's intercalation. That's a pretty big list, but let's look at them one at a time. Sometimes deoxyribonucleotide bases can adopt alternate conformations by tautomerization. And the change of conformation that we see here in adenine is caused by a migration of the hydrogen shown in red and a double bond. There was a double bond originally between the nitrogen at position 1 and the carbon at position 6, as shown in the diagram in the upper right. This double bond is now formed an immunotautomer. Because of this, the nitrogen at position 1 is no longer a hydrogen bond acceptor. It's a hydrogen bond donor, and for this reason, adenine in the tautomeric form can bind with cytosine because the pattern of hydrogen bond acceptor and donor is complementary. And so tautomerization is the migration of a hydrogen atom or a proton and a concomitant switch of a single bond and an adjacent double bond. In this diagram, we see the predominant forms on the left and the more rare forms on the right. And in each case, whether we're going from an amino group to an imino group in adenine and cytosine, or from a keto group to an enol group in guanine and thymine, these changes in configuration cause differences in hydrogen bonding patterns. Now, are you going to have to know the difference between a regular adenine and its tautomer? No, you're not going to have to know those structural differences. But you do need to know the concept, and you do need to know what tautomerization is and why it can cause mutations in the DNA. You also do need to know that the common forms for deoxyribonucleotide bases are in the amino form and the keto form, and the rare forms are the imino and the enol forms. Base pairing can also occur because of differences in hydrogen bonding between the syn versus anti conformations of the bases with respect to the sugars. Now purines, once in a while can be in the syn conformation, but most of the time, purines are in the anti-conformation. This is normal. Pyrimidines, on the other hand, are always in the anti-position. In the diagram above, we see syn-guanosine and anti-guanosine. You can see that in the anti-position, the bases are extending toward the center of what would be a DNA helix, whereas in the syn position, the base lies over the ribose sugar. In the panels on the bottom, on the left, we can see the normal hydrogen bonding between guanine and cytosine, and on the right, we can see the abnormal bonding between adenosine, which has flipped into the syn conformation, and guanosine, which is in the normal anti-configuration. In these configurations, the patterns of hydrogen bond donors and acceptors are such that adenine can bind with guanine. Base pairings can also be disturbed when water gets into the center. Here we see a water molecule in the center between a thymine and a cytidine, and this is shown by the purple arrow. This disturbs the hydrogen bonding that would normally form, and so thymine can now bind with cytidine rather than with adenine. Chemical mutagens can also cause changes in hydrogen bonding patterns. For instance, an alkylating agent that adds a methyl group to the ketone in guanine can prevent the hydrogen bonding that would normally occur at that oxygen, so we've lost a hydrogen bond acceptor. And because of this alkylation, there's steric hindrance for the formation of a CG base pair, and for this reason, O6-methylguanine will sometimes pair with thymine. One of the most well-known alkylating agents is alpha-toxin. Alpha-toxin is a toxin that's produced by a fungus that attacks peanuts. This alpha-toxin, on its own, can intercalate between base pairs, but an enzyme in our liver does a really nasty trick. When cytochrome P450 tries to detoxify alpha-toxin B1, it actually forms an epoxide, which is a highly reactive DNA-modifying agent. So, this activated alpha-toxin can not only intercalate between base pairs, that epoxide can form an alpha-toxin DNA adduct, and this is what we see in the lower left of this diagram. And so alkylating agents add carbon-containing molecules as adducts where they shouldn't be. Reactive oxygen species can also attack DNA bases. In this case, we've oxidized the carbon at position 8 to form 8-oxoguanine. In so doing, we've added another hydrogen bond donor at nitrogen 7. And so this phase is a perfect complement to adenine. Now another thing that can happen is the loss of hydrogen bond donors. This happens through the loss of an amino group. Chemical mutagens, such as nitrous acid, can cause oxidative deamination. Cytosine will lose an amino group to become uracil. We've seen this deamination before, and now because we have a hydrogen bond acceptor instead of a hydrogen bond donor, uracil will bind with adenine. Now adenine itself can become deaminated to form hypoxanthine. If you'll recall, hypoxanthine is the nitrogenous base that's found in inosine. Hypoxanthine can bind to cytosine because now there's a hydrogen bond acceptor at that ketone group and a hydrogen bond donor, and so for this reason, hypoxanthine can bind to cytosine. Now finally, there are some chemicals that can cause major disturbances in the DNA base pair stacking. Now thymine dimers are a type of pyrimidine dimer that is caused by UV damage. Ultraviolet rays from the sun cause bonds to form between adjacent thymines, and these are thymines on the same strand. And as you can imagine, this causes quite a disturbance not only to these nitrogenous bases, but also to the DNA backbone. Another thing that causes a disturbance to the DNA backbone is an intercalating agent. Intercalating agents are planar molecules that fit right in between the base pairs, and when they do so, they disturb the normal packing of the nitrogenous bases and also the DNA backbone. And in this diagram, we see ethidium stacked between the base pairs here. You might be familiar with ethidium as ethidium bromide. Ethidium bromide is an orange-colored stain that fluoresces under UV light and is used as a stain for DNA in DNA gel electrophoresis. So ethidium and molecules like ethidium are potent mutagens and are considered carcinogenic. So be really careful and wear your gloves with this stuff. Now the things that we've been talking about cause changes in DNA sequence. These can turn into genetic mutations if they're not caught in time before the cell actually divides. Once this happens, these mutations are passed on to a daughter cell, and from then on, they can be replicated and passed on to future generations. So there's a tight window of opportunity to catch those changes in sequence and repair them before they become genetic mutations that are heritable. Sometimes these changes in DNA sequence are such that they can actually prevent replication from occurring altogether. They can stall the replication machinery and cause it to fall off. Now, if mutations are severe enough so that they cannot be repaired in time, the cell might actually opt to undergo cell death rather than pass on those mutations to further generations. And this is a fail-safe to protect the body from such things as cancer. Let's take a look at the classification of mutations that can occur in DNA. These mutations, as we saw, can occur through chemical modification, through cross-linking, or through errors in replication. They can result in things like point mutations, which are a single deoxynucleotide change in the sequence. They can also cause insertions or deletions of deoxyribonucleotides, so adding more deoxynucleotides than is normal or taking some away. Double-stranded breaks can also occur in the DNA, and these double-stranded breaks can cause things like inversions, translocations, and ring chromosomes. An inversion is shown in A. This happens when there's a double-stranded break that causes that portion of DNA to be inverted and then reattached. These inversions occur on the same chromosome. Translocations occur when a piece of DNA is passed from one chromosome to another. Ring chromosomes happen when the double-stranded breaks anneal to one another in the same chromosome to form a ring-like structure. Now, genetic mutations that occur as a result of one change in the DNA sequence are called point mutations. These are also called single nucleotide polymorphisms, SNPs. This is a change in one deoxyribonucleotide. These can result in a change in phenotype, the way an organism looks or behaves, or it can have absolutely no change in phenotype and be silent. These point mutations, although small, can have a large effect on function. If you recall, it is only a point mutation that gives rise to sickle cell anemia, for instance. And so just because it's one deoxyribonucleotide that's been changed, there can still be very large effects in phenotype. These point mutations can occur in a non-coding region and give rise to splicing errors or altered regulation of the DNA, either making more of the product or less. Or these mutations can occur in the coding region. They can give rise to amino acid substitutions. These are called missense mutations. These can either be conservative mutations, and we saw these before earlier on in the semester. Conservative mutations exchange one amino acid for an amino acid with a similar function due to a similar shape or charge or degree of hydrophobicity. Radical mutations are just what they sound like. You're substituting one amino acid for another that's hugely different. Now, missense mutations can also give rise to mRNA splicing errors, so taking out a splicing region where there should be one, or making a splicing region where there shouldn't be. Now, when there's no change in the amino acid in the coding region, this is because of the redundancy of the genetic code. There are some amino acids that can be encoded by several codons, and so therefore, a change in the DNA sequence does not always equal a change in the amino acid sequence. And finally, a point mutation can introduce a stop codon, which can cause the truncation of the protein product. And this is called a nonsense mutation. Now, there can be larger changes in the DNA sequence caused by insertions or deletions of deoxyribonucleotides. These insertion or deletion mutations are called indels. They can be anywhere from one base pair removed or inserted, or several hundred base pairs removed or inserted. Now, a simple insertion or deletion is what happens when 3 or multiple of 3 deoxyribonucleotides are added or taken away, and this is because it causes no frame shift. Remember, if you're taking out a 3 deoxyribonucleotide chunk, you're taking out a codon. However, if you take 4 or 5, then you have a problem because the DNA shifts downward and you've changed every single codon that comes after that deletion. Same thing with an insertion. If you insert 1 or 2 deoxyribonucleotides, then you shift everything that comes after it up. And so you change the wording of the message. So for instance, if the original codon is ATG, CTG, ACA, GCG, this encodes methionine, leucine, threonine, and alanine. If you take out one nucleotide, if you take out that G that's between the T and the A, you end up with a frame shift. And the first 2 amino acids are just fine. You have methionine and leucine just like before. But now you have a frame shift right after that. You have glutamine and arginine. So you went from an alanine to an arginine in that 4th position. And that, my dear, is a really radical substitution. Uncharged and charged, small and big. Not such a good state to be in. The insertion we see next is an insertion of 4 deoxyribonucleotides, GATT. And so here we have methionine, and we go right into the problem, aspartate, serine, and aspartate. So now we've put in negative charges. This could be a problem. And no telling what anything else is downstream. And so how are these problems repaired? We're going to look at the repairs that happen in E. coli because they're far more simple to understand, and in many ways, the repairs that happen in eukaryotes are quite similar. They're just a lot more complex. We're going to look at the 3 basic methods of repair. These are mismatch repair, nucleotide excision repair, and base excision repair. And we'll start with mismatch repair. Now, as the polymerase goes by and synthesizes DNA, some mutations can be introduced and can cause mismatches in the DNA. Now, lucky for us and for the E. coli, these mismatches can be recognized. But the thing is, here we see a G and a T, which is an obvious mismatch. There should either be a C there to pair with the G, or there should be an A there to pair with the T. So something has gone wrong, but how do you know which is which? How do you know which deoxyribonucleotide you should remove and replace? Should you replace the G or the T? The good thing is that there's a way of finding out. The parental strand is methylated, but when the new strand is synthesized, it's not methylated yet. And so we know that the new strand must be the one with a problem, and therefore, that's the one we want to change. Now, the mismatch repair mechanism relies on the mute proteins, mute S, mute L, and mute H. First of all, the problem is going to be identified by mute S. Mute S and mute L are going to form a complex together. And then mute H is going to find the next methylation site. It's a GATC sequence that's closest to the mismatch, and it's going to bind there. As mute H is bound there at the methylation site, it can recognize which strand is methylated and which is not. But in order for it to work, it has to be activated by mute L. And so Mut S and Mut L are going to bend the DNA around, and Mut L will contact Mut H and activate it. And then Mut H is going to nick the non-methylated strand, the daughter strand, the one with the problem. Then the bases between the methylation site and the problem site, the mismatch, are going to be removed. Now this happens through the action of various exonucleases. It depends on the position of the methylation site in relation to the mismatch repair site, whether this portion is going to be removed in the 3' to 5' or the 5' to 3' direction. So we're not going to get into the particulars of this. Just know that the next step is the excision of nucleotides between the methylation site and the mismatch. The next thing that's going to happen is we're going to synthesize the new DNA and resolve that problem. This happens through the action of Pol3, which is the major replicative enzyme, and the breaks in the DNA are going to be sealed by DNA ligase. And as we remember, this is an enzyme that requires ATP, and this is a breakdown that you can use as notes to study by. Now, thymine dimers are an unusual thing. This happens when two adjacent thymines bond together, and it's usually a result of UV damage. Most cells in most organisms have an enzyme called DNA photolyase that can use the energy of the sun to break these bonds to release individual thymines. But placental mammals like us can't do this. We don't have this enzyme. But one of the things that we do have is the ability to resolve this during transcription. The transcriptional machinery pauses when it hits one of these, and an enzyme comes in whose job it is to put A's across from these two T's. In this way, the DNA template can be used accurately, because this enzyme knows that if there's a problem like this, the ribonucleotides that must be used to make this RNA have to be two A's. And so that's one way we get around this. Now we also use something that's called nucleotide excision repair, and so does E. coli. And we're going to look at the E. coli mechanism here. The enzymes actually stand for UV repair, UVR, ABCD. UVRA is involved in recognizing these thymine dimers, and UVRB and C are the exonucleases that are going to cleave to one side and to the other side of this thymine dimer. And so now we have a break upstream and we have a break downstream. Next comes UVRD. That is a helicase that's going to drive the unwinding of this small portion of DNA that includes the thymine dimer. RecJ is an exonuclease that's going to help to degrade this DNA, and the single-stranded DNA that's left is going to be covered quickly by SSB to prevent double-stranded DNA from forming when it shouldn't. Then DNA Pol1 is going to synthesize the DNA, and again DNA ligase is going to seal the gaps. And so that's nucleotide excision repair, NER. Now base excision repair is also pretty easy, and this happens often when cytosine spontaneously deaminates to form a uracil. Now, we all know that there is no uracil in DNA. There's thymine instead. And so if there's a base that looks like a uracil in there, it has to be gotten rid of because it's actually a cytosine gone bad. So the base excision repair machinery gets to work, and the enzyme that does this is uracil glycosylase. It's going to cleave the glycosidic bond between the uracil base and the ribose sugar. The incorrect base is actually going to be flipped out, and the glycosidic bond is going to be cleaved. In this way, the base is released, but the sugar phosphate backbone is still intact. This results in what's called an AP site. It's either apurinic or apyrimidinic. In the case of uracil, it's an apyrimidinic site. Now enzymes are going to excise the rest of this nucleotide, and DNA Pol1 is going to insert the correct nucleotide according to the template. and that's base excision repair. Now, other sorts of mutations can also occur, and this happens when there is something called a trinucleotide repeat. This is seen in Huntington's disease and also in Fragile X syndrome. In the case of Huntington's disease, the gene that gives rise to Huntington is involved, and the trideoxynucleotide repeat is CAG. It's called a polyglutamine track because CAG gives rise to glutamine. During replication, a portion of this DNA slides and forms a loop, and in this way, it becomes extended, so that there are more and more CAG repeats as this cell divides. Now the thing is, 6 to 31 repeats of CAG, that's a normal status. 36 or 82 or even more, then the individual is going to be affected. These CAG repeats can expand as the generations pass. And if you are affected by that particular genetic mutation, you can look to your ancestors and see when they developed the disease and how bad the disease was. You have a chance of being symptomatic earlier and having more severe symptoms than your ancestors. This is called anticipation. Now, as we said, double-stranded breaks are more severe lesions, and they can be repaired by DNA recombination. In addition, recombination also allows new combinations of genetic material to be formed through the physical exchange of DNA. This is known as homologous recombination that occurs between two very similar sequences. Sometimes we can even have recombination between non-homologous sequences. Why do we do this? This allows for DNA repair, an increase in diversity in offspring. It allows us to generate antibody diversity, and it even allows organisms to introduce new genes into the genome through things like transposons, for instance. Homologous recombination uses the enzymes REC-BCD, REC-A, and RUV-ABC. Now, in REC-BCD and REC-A, the REC stands for recombination, and RUV stands for repair of UV damage. So let's look at what happens. The first thing that we have to do is we have to identify the double-strand break, and this happens through the action of REC-BCD. Now, REC-BCD is a complex that includes helicase functionality and also endonuclease functionality. REC-BCD generates a 3' overhang and coats it with REC-A, and begins to invade the double-stranded DNA of its homolog. In this diagram, the homolog is shown in red. The invading 3' strand is shown in blue. Once the REC-A coded 3' end has invaded the homologous DNA, it finds the homologous sequence, and then DNA polymerase begins to polymerize DNA to replace what's missing, and then DNA ligase seals the breaks. The next thing that happens is something that's called branch migration. We have generated two crossover points, and these migrate along the DNA to create a more stable interaction between these two homologs in preparation for cleavage. Now, in order to separate these 4 strands of DNA into 2 double-stranded molecules of DNA, branch migration is facilitated by the enzymes RUV-A and B. The enzyme RUV-C cleaves the crossover points in order to generate 2 independent double-stranded portions of DNA. This marks the end of Lecture 36. Have a great day.